So we're going to do that pretty much every week, not necessarily every class. So some classes will be like Mondays was, where we'll take breaks, but we won't actually go to breakouts. But some classes will be like that, where there will be time for you to work together. This week it was mostly geared towards learning, solving the problems for the problem set, which is appropriate. Some weeks it will be quizzing each other on anatomy to get ready for an anatomy quiz or an anatomy part of an exam. Now remember, this week you've got a problem set due on Friday, which you were just working on, and then you have an anatomy quiz due on Monday. Very fast, I understand. So you'll learn more anatomy in lab this week. You'll spend 2 hours doing anatomy in lab, and then using the coloring book to reinforce and learn your anatomy for the anatomy quiz. The anatomy quiz is not on Moodle, so the problem set is on Moodle, as you now know. The anatomy quiz is on paper. You'll pick that up at the CST desk, sit down and take it, and then turn it in there. Okay, so let's talk about synaptic and action. Synaptic potentials and action potentials. Some of the questions on the problem set you might have deferred to, looking at it and saying, I think he's going to cover that in class now, which is what I'm going to do. So we're going to talk about synaptic potentials and then two types of synaptic potentials: excitatory postsynaptic potentials and inhibitory postsynaptic potentials. Sodium channels and action potentials related to sodium channels and potassium channels. So, cellular anatomy, dendrites, where receptors are bringing information into the neuron, thousands of them per cell. Soma, axon, myelin, synapse, the connection between one cell and the next cell. And so the synapse can either be excitatory or inhibitory. Each synapse is either excitatory or inhibitory. But this cell is receiving thousands of input, thousands of synapses all over it. We're only looking at 4. Really going to focus on 1. You have to remember that each cell is receiving thousands of inputs. Some of them are excitatory and some of them are inhibitory from the thousands of different cells that are connected to the cell. Synaptic potentials are small, local changes in the resting membrane potential of the receiving cell. Action potentials are going to be large, propagating signals, mostly in the axons. So we've talked about receptors. Let's focus on a subtype of the receptors, not GPCRs, although GPCRs can affect membrane potential. Let's look at the ionotropic receptors, or ligand-gated ion channels. So if the neurotransmitter released at the synapse by this cell, if that neurotransmitter activates a ligand-gated sodium channel, then the delivery of positive charge to the inside of the cell reduces the inside negativity, makes it less negative, and it's a depolarization. And one of the questions on your problem set is related to acetylcholine receptors, which are not selective for sodium, but because they allow sodium to flow in, they cause a depolarization. You've discovered that by solving, I forget, one of the problems in question 2. If, on the other hand, the neurotransmitter released at this synapse by this cell, if it's a different cell, is if the neurotransmitter activates a chloride channel, chloride flows in, down its chemical gradient. Chloride is high on the outside, low on the inside. So even though it's negative here, chloride flows in down its chemical gradient and makes the membrane potential more negative. So depending on whether the cell is receiving this type of neurotransmitter or this type of neurotransmitter, activating this type of receptor or this type of receptor, we can get a small local depolarization, which is called an excitatory postsynaptic potential. Or, let's skip this part. Well, no, I don't want to skip it too much. So, what I mean, it's small because it's just that little synapse. And it's local. The flow of sodium is affecting it's affecting the voltage in a small amplitude, and it's affecting it in a small area, small zone of the cell. So it is small and it is local. Question, Vincent?
Yeah, so I was wondering if a lot of these local synaptic potentials could possibly build into an action potential?
Exactly, that's where we're going.
Okay, cool.
We're going to talk about summation, both temporal and spatial summation. But yes, they do build. So a small local depolarization, which dissipates as you move away from the site of the synapse. If, on the other hand, going back here, if on the other hand we open we release this neurotransmitter, GABA is the example in your problem set, and it opens up a chloride channel, then we get an inhibitory postsynaptic potential that takes the membrane potential away from threshold, a hyperpolarization. So the cell is receiving stimulatory input and inhibitory input all the time. Thousands of neurons connected to a single cell, causing EPSPs and IPSPs. The release of a small amount of neurotransmitter will produce a small EPSP, if it's an excitatory neurotransmitter, and the release of more neurotransmitter will cause a larger EPSP. The point here is that synaptic potentials are graded. They can be small or they can be big. Graded just means there's variability in their amplitude, depending on whether the stimulus was small or big. So EPSPs can be graded. IPSPs can be graded in the same way. This is just a point making the same point here as is made here. If we measure the depolarization out here, it is small relative to the depolarization right here next to the channels that are triggering the depolarization. Okay? So it's all happening all the time. They're small and they are local and they are graded. Thousands of them receiving depolarization and hyperpolarization stimuli all the time. And it is at this area, this is the axon cut off. Here is a dendrite cut, here's a dendrite cut, here's a dendrite cut, here's the soma. So we're just seeing we're seeing the soma and parts of dendrites and part of the axon. All of these little blobby things are supposed to be the synapses of presynaptic cells making synapses on this yellow cell. the action potential is going to happen in the axon. The part of the axon that decides whether to fire an action potential, and if so, how many, is the beginning segment, the so-called initial segment of the axon. That's where the action potentials are initiated. We'll get to action potentials in a minute. But if we depolarize, so here's a question. Do all synapses influence the initiation of action potentials equally? Why not, Vincent?
Well, I'm thinking because like synaptic potentials are localized, then the ones that are closer to that beginning area of the axon would have more influence over the action potential creation.
Exactly. The ones that are closest to the action potential initiation zone have the more impact, but there is summation going on all the time. So getting back to Vincent's other point, here are two excitatory postsynaptic potentials separated by time. Here are two where the time separation is less. They are almost simultaneous. They're closer together. And so we get some summation of those EPSPs, summation of the depolarization, because the stimuli are close together in time, and that's temporal summation. Similarly, we could have one synapse causing a EPSP here, and another synapse, let's say from a different cell, causing an EPSP here. There's going to be spatial summation, especially if they are closer together than cartooned in this case. So if this synapse produces this EPSP, and this synapse produces this EPSP, and both of them happen at the same time, you could say that because this is two different synapses, we call it spatial summation. So temporal summation is when one synapse fires on top of each other, and spatial summation happens when multiple synapses are firing, multiple synapses are causing EPSPs simultaneously. And so you can see we could have temporal summation and spatial summation going on at the same time, getting the cell to threshold, adding up, and triggering an action potential. Okay, questions about synaptic potential summation? This is where we're going now, is to talk about action potentials. Of course, there's summation of inhibitory and excitatory potentials as well. If we have a bunch of excitatory EPSPs, but we also add in an inhibitory EPSP, the summation of a hyperpolarization with a depolarization results in less depolarization, a smaller, in this case, they're completely balanced. And you see no EPSP or IPSP. But that doesn't. It just means that the IPSP can summate with EPSPs and reduce the impact. So you might be asking, do G protein coupled receptors cause EPSPs or IPSPs? Well, it all depends on what's downstream from the receptor. The receptor itself does not change the membrane potential because no ions flow through the receptor. It's not coupled. A GPCR does not do this. But there are channels that are coupled downstream of G proteins. So GPCRs can trigger IPSPs or EPSPs depending on what their alpha subunits work with, or what their beta gamma subunit, what their beta gamma complex is coupled to. It depends. So the cartoon that you have is that what's downstream from a GPCR are kinases. That's not always true. Sometimes there are channels downstream from a GPCR. Chloride channels, potassium channels, even sodium and calcium channels can be downstream from GPCRs. By downstream, I mean the G protein affects the channels, or maybe the kinase that the G protein receptor is coupled to. The kinase phosphorylates a channel and changes its activity. Okay, so now let's talk about action potentials. Action potentials and synaptic potentials are the way that information is coded in your nervous system. And we're going to see that in the next section of the course. So here's a stimulus that gives this patterning of action potentials. A bigger stimulus gives a different patterning of action potentials. A bigger stimulus still gives a very different frequency of action potentials. So while synaptic potentials are graded, you'll notice that action potentials are not. They all have pretty much the same amplitude. Action, that's what, when people talk about, and I think your book talks about the all or none feature of an action potential, that's what they're talking about. You either are firing an action potential of a given amplitude, or you're not. So what is coded, so synaptic potentials, what is coded is the amplitude. Small stimulus, small amplitude. Large stimulus, large amplitude. What's coding information in action potentials is the frequency and the pattern. So a small stimulus gives a low frequency, or just a few. A large stimulus gives a high frequency, or a lot. It's not, the amplitude is not changed, it's the frequency that is changed. So let's talk now about what some of the jargon of an action potential. So here's EK, here's the equilibrium potential for potassium down around minus 90. Here's the equilibrium potential for sodium up around plus 60 or 66 or whatever, up there somewhere. The resting membrane potential is negative, but it is not usually all the way to the equilibrium potential for potassium, but it's down here somewhere, minus 60, minus 70, minus 80, minus 90, depending on the resting permeabilities that your problem set asks you to work on. If this cell receives EPSPs that summate to threshold, once it reaches threshold, which we'll talk about in a moment, it fires an action potential. An action potential is a large, brief depolarization. So in this cartoon, this is only about 1 millisecond. So this thing here is the same as this thing here, because this time base, you have to pay attention to the time scale, this is 200 milliseconds. So this is 200 times longer than this. And that's why on this scale, an action potential looks like a spike. But if we blow this up and look at it expanded on the time base, it looks like this. Okay? So they're the same thing on a very different time scale. This hump, when we compact it down into 200 milliseconds, becomes a spike. Very tall, a large depolarization, and a very brief depolarization. 1 millisecond in duration, 1 millisecond in duration. There is you can talk about the upstroke of an action potential, the depolarization phase, and then the repolarization phase. Action potentials are large depolarizations, and they're very brief depolarizations, so there's a phase of repolarization. After the action potential, you'll see, and I think you had to work on this in the problem set, there's a region where the membrane potential goes past resting to a hyperpolarized voltage, and then it settles back down, back up to rest. Do we see hyperpolarizations in this actual data? Yeah, I mean, so, well, no, not really, because, well, there's one. There's a hyperpolarization. There's a hyperpolarization. It's settling back to rest. What causes action potentials is a feedback loop caused by voltage-gated sodium channels and voltage-gated potassium channels, working in a sort of concerted, coordinated way. So let me unpack some of that language. Voltage-gated sodium channels. These are channels that are opened by depolarization. And now you're thinking, okay, they're opened by depolarization. If they open, sodium flows in, so they cause depolarization. So there's automatically a positive feedback cycle. They are opened by depolarization and they cause a depolarization. There's a positive feedback loop that causes the membrane potential to go from something negative around the threshold to zoom up towards the equilibrium potential for sodium because of this positive feedback loop. And then after that, that positive feedback loop is interrupted, and voltage-gated potassium channels are activated to cause the repolarization. So let's go through these loops, these positive feedback loops. Okay, what I want you to remember is that voltage-gated sodium channels are opened by depolarization, and then they cause more depolarization due to the sodium influx through them. So here is the stimulus. This is what's going to get us into this loop. Local depolarization. A synaptic, an EPSP. Right there. It opens voltage-gated sodium channels. Sodium flows in. Causes a depolarization. Which does this again, right? And we run this loop so fast that in something like 1/10 of a millisecond, it only takes, it's much less than a millisecond to go from threshold to the peak, because we run this loop so fast. And it's fast because voltage-gated sodium channels are fast. They're very quick to respond to depolarizations. They have evolved to be rapid detectors of depolarization. And they respond, and they open fast. And ion flux. Ion flux is fast. Ion flow is fast. So this feedback loop runs very fast, and in less than a millisecond, we go from here to here. So let's talk about this as an all or none. and talk about threshold. Threshold is the voltage depolarized from rest that is enough, that opens enough sodium channels to fire an action potential. If the small depolarization, remember there's small local depolarizations of the EPSP, if the small local depolarization is not big enough, it is lower than threshold, the membrane potential settles back down, it just decays back to where it started from. So there are going to be sub-threshold EPSPs and supra-threshold EPSPs. You go above threshold, you fire an action potential. If you don't, you don't. The fact that this feedback loop, once enough sodium channels are opened by the beginning to cause this feedback loop to go around and around, and it opens all of the sodium channels, that's what makes action potentials all or none. You either open up enough to open them all, or you don't. You never open up some of them, unless there's a toxin in there. And I think I gave you a problem where there's the fugu toxin. That's a toxin. That's not physiology. That's having a drug. Physiologically, this peak is determined by how many sodium channels you have to be opened by the positive feedback loop. If you only have a few voltage-gated sodium, if you have a smaller number of voltage-gated sodium channels, what is that going to do to the amplitude of an action potential in that neuron? It would make it small. But that neuron is going to have a defined action potential peak defined by the number of sodium channels that it expresses. If you block some of them, I think you've got a question that would say, think that through. If you block some of those sodium channels, what is that going to do to the action potential? That's a problem. I'm not going to give you the answer. Sodium channels have a very interesting function that after they open, each of them molecularly, after it opens, it undergoes a slower conformational change into an inactivated state. So inactivated means they're not open. Now, so you have to be very careful. I have to be very careful here. You could say, well, inactivated is closed. And I would say, no, closed is what happens before the action potential. And inactivated is what happens after the action potential. And there's a difference. Before the action potentials, the channels are closed and are available to open. After the action potentials, the channels are inactivated and cannot reopen. There's a difference between the closed state and the inactivated state. And then you'll say, well, if they can't reopen, then how do you ever get another action potential? And I'll answer that in a moment. So the sodium channels go through this loop, get to the peak, and then inactivate. What allows them to recover is during repolarization, they can recover from inactivated back to the closed. So this is a state diagram. It shows that resting voltage-gated sodium channels are closed. At the resting membrane potential, most of them are closed. Very, very few of them are open because they have not been stimulated to open. They could open, but they have not received the stimulus. When they are depolarized, they open. The channels undergo a conformational change from open to inactivated. In a depolarized voltage, if the voltage stayed depolarized, the channels would stay inactivated. But they don't stay depolarized. We're going to talk about the potassium channels that cause repolarization. And so when the channels are down here, inactivated, they can recover from the inactivated state. So the inactivated state happens. Repolarization is what allows them to recover.
So is it the repolarization itself that causes the inactivated channels to become closed and then be able to open again, or is there some other factor?
Say that again, I want to make sure I understand.
So like, what is the specific factor that causes the inactivated channels to not be activated anymore? Is it the repolarization or?
Repolarization. So the question is, what is the specific factor that allows sodium channels to not be inactivated anymore? Did I say that right?
Yeah.
And the answer is repolarization and time. It takes some time, a few milliseconds. Not hours, not minutes, not seconds, milliseconds. So the sodium channels are closed, open, still mostly open, inactivated. They have transitioned to the inactivated state. They would inactivated, still mostly inactivated, but now we are repolarized and we give it some time and they are recovered back to closed. Zoe, did that help?
Yeah.
So repolarization and time recovers voltage-gated sodium channels from the inactivated state back to the closed available state. And from closed, they can reopen and fire another action potential. But from inactivated, they can't. In addition to this loop, depolarization also opens voltage-gated potassium channels, but they have an intrinsically slow kinetics. So the same depolarization that opened sodium channels opens potassium channels, but with slow kinetics. So the voltage-gated sodium channels open first and then inactivate. The voltage-gated potassium channels open while the voltage-gated sodium channels are inactivating. They're slower. They're intrinsically slower. It takes a few milliseconds as opposed to a few microseconds to get the voltage-gated potassium channels to open. So the same depolarization that caused the positive feedback loop opens voltage-gated potassium channels that cause outflow of potassium, and it accelerates repolarization. Now, why am I being coy about this accelerates repolarization? If we didn't have, let's just think this through. If we didn't have any voltage-gated potassium channels, but we had perfectly normal voltage-gated sodium channels, and we opened them, would the membrane potential repolarize? Sarah, why?
You still have the resting potassium channels.
Right there. You still have the resting potassium channels. So if the sodium channels inactivate, and we still have those resting potassium channels, we will still get repolarization. But if the sodium channels inactivate, and then we also bring into the equation voltage-gated potassium channels, we repolarize faster. So we have fast, very fast activation of voltage-gated sodium channels, which then inactivate. We have slower activation of potassium channels, some of which inactivate and some of which don't, and that's an upper level issue. So for this course, let's just think about the potassium channels that activate slowly and then close when the membrane repolarizes. So these are channels that are opened by depolarization and closed by polarization or hyperpolarization. That's good enough for us. So that's synaptic and action potentials. Let's do some clickers together for 5 or 10 minutes, and then we'll call it a day. Well, no, we do have lots of other things to do. We'll go have lunch. So let's do this as a clicker here. You know that sometimes I say we want to do clickers as homework, but sometimes when there's time, we'll do them together. Let's do the first one. During the after hyperpolarization. So what is the after hyperpolarization? This is the after. What is it after? It's after the action potential, and it's a hyperpolarization. The name is pretty descriptive. After hyperpolarization. During the after hyperpolarization, which of these are true? And let me be specific. Let's say right here, the maximum after hyperpolarization. Not way out here, right here. Show me your answers. Okay, I see some correct answers and some not correct answers. The sodium channels have transitioned into the inactivated state, and the potassium channels are open. Either the resting ones or the voltage-gated ones are still open at that point. And that's what allows the maximum after hyperpolarization. Vincent?
Are they, so I said C and D, but are they not also closed while they're inactivated?
Which ones? The sodium
The
channels?
sodium channels.
No. They can either be closed, open, or inactivated.
Okay.
Well, each channel can either be, let me be quite careful here. Each channel can either be closed, open, or inactivated. The population can be spread out, but at this point in time, there has not been enough time to allow the voltage-gated sodium channels to recover. So they are mostly inactivated. And as we move out here, they move from, the population moves from inactivated to closed. But here, they're mostly inactivated. Here, voltage-gated sodium channels are 50-50. And here, voltage-gated sodium channels are mostly closed. Okay, what is required? I think this is Zoe's question. What is required for sodium channels to recover from inactivation? So I think I gave you this one, so we can do this one quick. Show me that you were listening. Okay, good. B and D. Okay? At the peak of the action potential, let's talk about flow. Which way are ions flowing at the peak of the action potential? So by peak, I mean here. Which way are ions flowing? Sodium and potassium. Okay, I'm seeing a lot of fully correct answers. A and C. Sodium is flowing in, that's what causes the depolarization, and potassium is flowing out. That is negating the depolarization, and eventually this is going to win as the sodium channels are inactivated. Right? But potassium, so at this point in time, there is sodium permeability, and the voltage is below E and A, so there's inflow of sodium. And at this point in time, there is potassium permeability, and we're way above EK, so there is outflow of potassium. So at that point in time, both of these are true statements. So your challenge is to put all this into your head, to use the problems that we didn't do in class to reinforce your knowledge, to use the problem set to reinforce your knowledge, to use the coloring book to learn anatomy, which we will reinforce in lab, to work approximately 3 hours a day doing all that stuff, to asking me questions if you get hung up, and you should not feel, I wish you would not feel inhibited about asking for help. You can ask for help on the problem sets, you can ask for help on the clickers, you can ask for help on a concept. That's your job, is to ask for help. You can't ask for help while you're doing a quiz. Well, you can ask for clarification, but you can't ask for an answer. All right, so today is one of those few days where we're going to be done early. Remember that sometimes I let you out early, even sometimes I don't. So, have a good afternoon. I'll see some of you Thursday evening in lab. Remember, lab is not here. Lab is in Stanley 151. And I'll see the rest of you on Friday morning in lab. There's no class on Friday because we're in lab. Some of us are in lab.